Hello students, welcome to Trunkie Academy. We were studying about the biotechnological principles and biotechnology, okay, and we studied about the different process that is involved in the biotechnology. And today we will study about the regulation of after biotechnological process. We also study about the gene Griffith experiment, and then we studied about the Harsa and Chase experiment, okay, and that was under the search of genetic material. So after knowing that the genetic material, we now will study about the regulation of gene expression. Okay, so try to understand. This is one of the most important topic. Okay, and it's the most important concept when you go to your you get into the medical college. This concept will be one of the very important. Okay, very among the very important topic. Regulation, how the gene is expressed, expression is regulated. Okay, this is an important topic. Now, what do you mean by regulation of gene expression? Regulation of gene expression refers to a variety of wide sequence of events that may occur at various levels. Okay, it may be expressed at different levels in case of eukaryotic and in case of prokaryotic. Okay, so regulation of gene expression refers to a variety, a very wide sequence, a very wide sequence of events that may occur at various levels. So, like we see, considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. Okay, so the main aim and objective of this expression is to produce. For example, if you consider this that expression, gene expression, or aim and objective expression is the formation of a polypeptide. So this formation of polypeptide can be regulated. In every case, yeah, this is going to produce polypeptide, but regulation of that polypeptide formation, okay, can be at different several levels. Okay, regulation of that formation of polypeptide chain can be regulated at different levels. Okay, so in eukaryotes, the regulation could be exerted at transcriptional level, formation of primary transcript. It may be at the level of transcription. Okay, when the transcription, at that time, this in eukaryotic cells. Okay, so it will be the transcriptional level, or it can be processing level regulation of splicing. Okay, then transport of mRNA from nucleus to the cytoplasm. When mRNA is being transported from the nucleus to the cytoplasm, carrying the message from the DNA and going to the ribosomes, so that time also it can be regulated, or at the translational level. Okay, where the translation of the mRNA is going on. So these are the 4 different levels where the regulation of the gene expression takes place. Okay, can take place. So now the genes in a cell are expressed to perform a particular function or a set of functions. Okay, so what we observe that a particular gene, okay, that is responsible for expressing a particular substance or product, okay, and that is responsible for a particular function to express. Okay, so genes in all cells are expressed to perform particular function, okay, or a set of functions. Okay, so chromosomes, you can see the nucleus, there is chromosomes, and the chromosome there is a gene, right? And for example, an enzyme called beta galactosidase is synthesized by Escherichia coli. So Escherichia coli are the bacteria that has a kind of gene called beta galactosidase in their chromosome or in their genetic material or DNA, which is responsible for producing the enzyme called beta galactosidase in the cell of Escherichia coli bacteria. Okay, so that particular gene, which is responsible for producing beta galactosidase enzyme, that will produce beta galactosidase. Because one particular gene will have one responsibility to produce this particular enzyme called beta galactosidase. So try to understand this. This is very important concept. It is used to catalyze the hydrolysis of a disaccharide. So for what this enzyme is required in bacteria, what they do with this enzyme, this particular enzyme produced in the cell of the bacteria, okay, with the help of a particular gene, okay, what they do with this enzyme, it is used to catalyze, break down the disaccharide lactose molecule, okay, into galactose and glucose, so that they can use the glucose as a source of energy, or glucose and glucose as this monosaccharide unit, or this monomeric unit they can use for the production of energy. Okay, when they are growing in the medium where there is lactose is present, okay, but no glucose and lactose is present. So what they do, they need to break down this lactose with the help of the enzyme called beta galactosidase, which is expressed, okay, and directed. The production of this particular enzyme, beta galactosidase, is directed by one particular gene, okay, that is present in the genome of the bacteria, right? So this lactose, with the help of this beta galactosidase, are broken down into two monomeric units called galactose and glucose. Okay, so there is a kind of you can see linkage. Okay, beta 1 for glucose. You can see galactose. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of enzyme beta galactosidase. So this is very important. Now, the gene which is responsible, try to understand this one, gene which is responsible for producing this beta galactosidase, this will not always produce this enzyme. Okay, this will not always direct the production of beta galactosidase enzyme. This will be active. This will become active, or this will direct the synthesis of this particular gene which is responsible for production of beta galactosidase. This particular gene will be active and direct the synthesis of this enzyme only when it is required by the bacteria, only when they will have this, like they are growing in the lactose medium, or the medium in which lactose is present, or when this bacteria do not have lactose around them to be utilized, for then this will no longer require the synthesis of enzyme beta galactosidase. So hence the gene, okay, which is responsible for directing the synthesis of beta galactosidase, that will remain switched off, that will remain inactive, okay, and becomes active only when there is lactose present around them. There is lactose present around the Escherichia coli cell. Okay, so the development and differentiation of embryo into adult organism is a result of the coordinated regulation of expression of several sets of genes. Okay, you understand this point? The development, the differentiation of tissues, development of the glands, formation of the gland, okay, then differentiation of the different tissues, maturation of the gland, okay, then differentiation of the embryo into the adult, into the fetus, then infants, then adults. Okay, all these also coordinated by the regulation of expression of several sets of genes. Several sets of genes are directing, they are regulating this process, and then this differentiation and development takes place. Okay, from embryo to adult. Now, glucocorticoid receptors in the absence of glucocorticoid hormone. So this will be like this. Since there is no in the absence of glucocorticoid hormone, so this will be free. So as the glucocorticoid hormone will be there, it will bind to the hormone, and you can see the kind of gene expression. Okay, you can see the gene 1, gene 2, gene expressed at low levels. Okay, and when the gene expressed at high level, when the hormone is present, right? So this is how in the presence of the substance which activates the gene expressions goes on at high level. Okay, now we'll see with a very good example. Okay, in prokaryotes, control of the rate of transcriptional initiation is a predominant target site for control of gene expression. So in prokaryotic cell, okay, the control of rate of transcriptional initiation, okay, is a predominant target site. Okay, is a for control of genes. So that is the for control of gene expression, the transcriptional initiation is the predominant target site. Okay. Now you can see here transcription taking place. Okay, from the DNA. Okay, then mRNA is formed. Okay, after transcription, and you can see the promoter region. Right. So from promoter region, You can see there's a kind of one enzyme text carries on this process of DNA polymerase, and then there is the transcription, the antisense strand. Okay, the two strands. You can see messenger RNA is synthesized here and forms the RNA after transcription. In our transcriptional unit or transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by the interaction with accessory protein, which affects the ability to recognize the start site. So what happens? RNA means RNA polymerase. Okay, whether to start the transcription or whether not to start the transcription. This is also controlled by or regulated by interaction with accessory protein. Okay, there is kind of another protein which is bind or which comes or which is present in the medium or organism. Then this activates the transcription, and then the RNA polymerase comes to a promoter, and then there is a transcription taking place, and the formation of RNA transcript is formed. These regulatory proteins can be both positively activators or negatively repressor. So try to understand. We'll see this in example, then we'll have a clear picture in your mind. Okay, we can go through the lack of around. After a few slides, we go through that. So the protein or the molecules which is responsible for starting the transcription, it can have the two kinds of effect. It can act in a positive way, activating or repressing by repressing the start of the transcription. So two ways: by activating the start of the transcription or by repressing the start of the transcription. So in two ways it may act depending on the molecules. The accessibility of promoter region of prokaryotic DNA is in many cases regulated by the interaction of protein with sequence term operator. So there is a kind of site in the DNA where we find a called site called operators. Okay, so that is an important region of that DNA that with the accessibility of promoter region of protein is in many cases regulated by interaction of protein with sequence term operator. So there is a kind of operator which is indirectly involved affecting the DNA transcription, and you can see here. This is a regulator gene, promoter gene, and operator gene. Okay, so when the operator gene will not allow the transcription from the promoter gene, if there is a kind of obstacle on the operator gene, so promoter gene, even it tries to transcribe, okay, it will not go on, okay, it will not be possible. Okay, so this yellow color shows that the structural genes, okay, expression of these genes lead to the formation of mRNA, transcription of which leads to the formation of that particular enzyme needed by the bacteria. Try to understand this. What is the accessibility of promoter gene is dependent, or you can say it is regulated by the interaction of protein with the sequence term. So the protein which is activated positively and repressing negatively, that is going to bind or they will represent the operator. So if operator gene has some kind of obstacle, this promoter will not move forward. The polymerase enzyme cannot move further. Okay, and there is no transcription of the structural genes, and there is no formation of mRNA from the structural genes, and hence there is no translation to be formed for the formation of the enzymes needed by the bacteria. Okay, the operator gene is adjacent to promoter element in most operons. Operon means a set of genes which is for one particular expression of the product or one function. So in most cases, the sequence of operator binds to a repressor protein. So we'll see that in example. Each operon has its specific operator and specific repressor. Now you can see the operator site and promoter gene, lake I, regulatory gene. So this lake I is called a regulatory gene. This will produce one particular kind of protein. This will be responsible for directing the production of a kind of special kind of the protein molecules with the help of the mRNA transcript. and this protein will then act as the active repressor. Then it will go to a operator site and it will bind to the operator site, then not allowing the RNA polymerase from the promoter gene to the structural gene, so that there will be expression of, or you can say there will be a transcription from the structural gene for the production of enzyme. So this is blocking the RNA polymerase. So the function of this, especially this operator gene, is to bind to the repressor protein molecule so that RNA polymerase cannot move further. Okay. So promoter is the site where the transcription starts. But how it will start if there is a kind of blockage, if there is a kind of hindrance, okay, by binding the repressor protein in the operator site. So that is one of the reasons. So for example, LAC operator is present in only in the LAC operon, and it interacts specifically with the LAC repressor only in case of here. So it is in case of LAC operon, regulatory I gene. In the absence of the lactose in the surrounding medium, okay, this LAC I is going to produce a kind of protein which is going to acting as active repressor. Okay, so now you see, sorry, LAC operon. Sorry, yes. Cell, right? Then you see, I just go through this and then you understand actually. Video will have some, give this idea, video will have. The E. coli LAC operon is an example of inducible cell, because the LAC, when lactose is present, that the what obstacle, what we call that, the repressor protein is removed. This is bacterial DNA, circular, and this is the repressor protein, and binds to the operator, not allowing the RNA polymerase to move. This is the lactose molecule. Now, this presence of lactose molecule will remove this repressor protein. Why? You see how you do this. This is a enzyme, beta glucoside. Another example, enzyme is produced like enzyme produced is the permease. This permease will allow the entry of glucose into the bacterial cell. Okay. So bacterial DNA. Then this is LAC operon. Okay. The control repressor. Now repressor is protein produced by the regulatory gene I. Okay. And this is bound to the operator gene. Okay. So now lactose molecules are available, and this lactose molecule will come to the this repressor protein and bind, and then it becomes inactive. Then it will move away. Then RNA polymerase can move further for the transcription, and this transcription of now structural gene taking place. This is mRNA transcript. The mRNA transcript has formed. Okay, and now this will direct the synthesis of protein, that is the enzyme called beta glucosidase and permease. Okay, so you see now it's the beta glucosidase is formed. Okay, again, let's see, the permease is formed, right? So this is how this takes place. Okay. Now you can see this. Now this permease is allowing the entry of this lactose molecule into the bacterial cell. It is more and more lactose, because if the lactose level in the growing medium, so this will be activated, and permease along with the beta glucosidase, permease enzyme will also be produced, and this will cause more and more the entry of the glucose molecule into the body. So what did you observe here? The main point is that you found that this repressor protein, which was bound to the operator site, was removed by binding the lactose molecule which entered the bacterial cell, and this caused the inactivation of this repressor protein and removal of this repressor protein from the operator site. Okay, so once all the lactose molecules were broken down, utilized, then what happened? Then there was the elucidation of the, then there was the again binding of this repressor protein to the operator site. The elucidation of the LAC operon was also a result of close association between geneticist Trunkie Jacob and a biochemist Jacob Snow. These are the two scientists. They did many experiments they did, Okay, and after many experiments with this bacteria, Escherichia coli, they found that how the lac operand works, how this expression of the gene takes place in prokaryotic cell. The Francois Jacob and the Jacobs Monod. Okay, so the scientists were first to elucidate the transcriptionally regulated system. In lac operand here, lac refers to lactose, a polycystic structural gene is regulated by a common promoter and regulatory gene. So genome, a set of genes, okay, which is responsible for a particular function, that is an operon, okay. So here, like polycystic, when we find see there are many genes involved for regulation of particular function. Like I promote, there is a PI, then there is a promoter gene, operator gene, leg Z, leg Z, Y, and A. These are the three structural genes responsible for directing the synthesis of enzymes called beta-galactosidase, leg Y, permease, and leg transacetylase. Okay, so these are the three enzymes found in the lac operon. Such an arrangement is very common in bacteria and is referred to as operon. This kind of arrangement in the DNA is found very commonly in bacterial genome, and it is called operon. A few such examples you can say lac operon, the tryptophan operon, HIS operon, ARA operon, okay, VAL operon. These are the different examples. Okay, the lac operon consists of one regulatory gene. Okay, the I gene here is termed as I, does not refer to the inducer. Rather, it is derived from the word inhibitor, not inducer, inhibitor, because this is producing the repressor protein molecule that is going to bind to the operator site, and it is not allowing the RNA polymerase to move further, and hence there is no transcription of the structural genes. Okay, so the promoter gene, then the operator gene, and they are forming the set. You can see that this is the RNA polymerase from the P1 and moving to the promoter site, but unless the repressor protein is not removed, this RNA polymerase are no longer functional, and there is no transcription. So the operand is in switch off condition. You can see the regions of coding, right? So the RNA protein is shown in yellow, and the diffusible regulatory protein. So LAC-I. This LAC-I is responsible for directing the production or synthesis of these regulatory, or you can say regulatory protein, or the repressive protein, or repressor protein, which is going to bind the operator. So the gene LAC-I expresses the mRNA, which is translated into repressor protein. This is the important point. Okay. I told you earlier also. So LAC-I, the gene codes for the repressor of the lac operon, and the gene codes for the beta-galactosidase is the LAC-Z or LAC-Z. The beta-galactosidase enzyme, which is primarily responsible for the hydrolysis of disaccharide, lactose, into its monomeric unit, galactose and glucose. Because bacteria need to break down this galactose into galactose and glucose so that they can produce energy from this monomeric unit. Okay, right? So the Y gene codes for the permease enzyme, as I told you earlier, they are responsible for allowing the lactose molecules to enter, more and more lactose to enter into the bacterial cells. Okay, so they increases the permeability of the of the cell to beta-galactosidase. Okay, increases the permeability of the cell to beta-galactosidase. So that is increasing the permeability, right? So the third one is the transacetylase enzyme. This is the enzyme. Not much of the function has been known about this. Okay, so this much on the hence all the three genes, they are working in a coordinated manner. The first, the LAC-I producing the inactive, or you can say the repressor protein, okay, which is going to bind to the operator site. Okay, once the allolactose, the lactose is slightly converted. Allolactose and lactose are very similar, quite similar, but there is very less difference. So allolactose is a lactose kind of substance, or it is a molecule that binds to the repressor protein and removes the conformational change in the structure of the repressor protein, and then that allows the removal of this repressor protein from the operator site, and hence the RNA polymerase is moving forward with the expression of the structural genes, and then translation of these mRNA the ultimately cause the formation of the enzymes, which are glycosidase, permease, and the transcriptase. The most other operants as well as the genes present in the operon are needed together to function in the same or related neuronal pathway. So, in other functions, operants also, okay, as the genes present in the operon, they need to function together, okay, in some related or metabolic pathway. Lactose is the substrate for the enzyme beta glucosidase and it regulates switching on and off the operon. Okay, when lactose is present in the medium, the bacteria is utilizing it. We want to utilize this lactose to break down this lactose into the galactose and glucose. Then this enzyme will be then the operon will be switched on. So that is called the switch on condition of that. Hence lactose is termed as inducer because this is inducing. The lactose or allolactose is also lactose is almost very similar kind of substance. When this lactose or allolactose is binding to the operator site, this is allowing the conformational change in the repressor protein, thereby removing the repressor protein molecule from the operator site, and then there is the RNA polymerase moving forward for the expression. Okay, so that's why it's called the inducer. So it's kind of inducible operon, where indeed this lactose is acting as an inducer molecule. In the absence of this preferred carbon source, such as glucose, if lactose is provided in the growth medium of the bacteria, it is transported into the cells through the action of permease, like I told you in earlier slides. Remember, a few low level of expression of lake operon has to be present in the cell at all times, otherwise lactose cannot enter the cells. So that is a very important point. Always there has to be some amount of lactose so that this operon is, because if it is growing in the medium where this lactose are present, okay, this the quantity of lactose that is present in the bacterial cell, that will activate switch on the operon system, and then the RNA polymerase starts transcribing the mRNA, and that will allow the formation and the synthesis of the enzyme, okay, needed for the breaking down the lactose molecule into this galactose and the glucose molecule, okay, then so they can produce the energy with the help of this monosaccharide units. The lactose then induces the operon in following manner, that is the repressor of the operon is synthesized at the all time and said to be constituted from the I gene. Okay, so the repressor protein binds to the operator and prevents RNA polymerase from transcribing the operon. Now, what is the main, like I told you earlier, operator site is just beside the adjacent to the promoter site. Okay, so RNA polymerase from promoter site cannot move, cannot start the transcription of the structural genes. Okay, unless and until that repressor protein is removed from the operator site. Okay, so when it is there, okay, and it is bound to the operator site, this repressor molecule is present in the time is called the switch off condition. In the presence of inducer such as lactose or allolactose, the repressor is inactivated. There is some kind of conformational change in the structure of that repressor protein molecule, and then it becomes no longer active to bind to the operator site and moves away from the operator site. Okay, so inactive direction of the inducer, and this allows RNA polymerase to get the access to the promoter and to the further structural genes, and then the transcription takes place and it proceeds further. Okay, so essentially the regulation of lac operon can be visualized as the regulation of enzyme synthesis by substrate. So main aim and objective here in the case of lac operon is to synthesize the enzyme for breaking down the lactose molecule into the galactose and glucose, so that bacteria can utilize it. The bacteria, Escherichia coli, when they are growing in the lactose molecule, they need to break down this lactose molecule into galactose and glucose for the production of energy. So essentially, the regulation of lac operon can be visualized as a regulation of enzyme synthesis, okay, by substrate. So remember, glucose or galactose cannot act as inducer. It is only the lactose molecule, allolactose, that acts as the inducer. Because once this lactose or allolactose binds to the repressor molecule, that changes the shape, or there is a conformational change in the shape of the repressor, and then thereby removing this repressor protein from the operator, okay, allowing the RNA polymerase to go further. Okay, so regulation of lac operon by repressor is referred to as negative regulation. Okay, so this is a lack of by repressor is called negative regulation, and by inducer allowing the regulation, okay, under the control of positive as well as what is beyond the scope of discussion at this level. That when we go to the higher class in the colleges, medical colleges, then we study about more in detail about this operon, because this is molecular biology, and at the level of molecular biology is not at all in the syllabus. Okay. So we'll go to the other point. That we'll see this in this diagram and the pictures. You see there is a kind of promoter site. Okay, the inducer is the gene I is the kind of inhibitor. Don't get confused with the I. It is not inducer. It is kind of inhibitor gene which is producing, or you can say responsible for synthesizing the repressor molecule. Then the repressor molecule is going to bind to the operator site, and this will not allow RNA polymerase for transcribing the structural genes. Okay, so this binds to the operator gene and prevents the RNA polymerase forming. So in the absence of inducer, this is in the switch off condition. As soon as the inducers enter or they bind to the operator, this repressor protein, which is bound, then this allows some conformational changes. Due to this change in the shape or the change in the conformation, this will move away. This will then just it is no longer active, and then slightly movable, and then I can say that it's not further binding furthermore, and then there is a transcription of structural genes. Okay. So then you can see the repressor mRNA, then there is a kind of inactive repressor, or then you can see the late mRNA, beta-glycosidase, transcriptase, and polymerase. These are the three enzymes, okay, produced by the set of these three structural genes, Z, Y, and A. So mainly the beta-glycosidase is needed because it is an enzyme which breaks down the lactose molecule into glucose and galactose. Okay. The gene Y is responsible for transcribing the permease enzyme, which is responsible for the movement of the lactose molecule and increasing. From the name itself you can understand, and you can just remember from the name. Okay, as it implies, you can say it increases the permeability of the membrane of the cell. Okay. So that's the reason it's called permease. And in case of transcriptase, the functions of this enzyme, transcriptase, is not much understood, but still some research is still going on. The day when this will be discovered, the functions will be understood. Definitely, this will also come into the syllabus. Then when we study about the detail function of the transcriptase. Okay, but till now it's not much understood. But somehow some idea. The scientists have got what is the role, but this is scope of the syllabus now. Beyond this scope of the syllabus. Now, in the presence of an inducer, this is happening. What's happening? Inducer, yellow color, that is binding the repressor molecule and making this inactive, or not allowing to bind to the operator site by changing its shape, or some kind of changes in the conformational change in its shape, so that it will no longer be active to bind to the operator site. Okay. Now we'll go through some important questions. Okay, very easy. But remember, this concept is very, very important. Regulation of gene expression is very important. This is one of the very common and one of the best example of the regulation of gene expression, the lac operon. That is it found in case of studied about the Escherichia coli prokaryotic cell, right? So an enzyme called beta-glucoside is synthesized by E. coli, yeast, or fungi. It is the E. coli. So here you can see that the lac operon model is proposed by the Hershey and Chase, R.D. Kornberg, Frederick Griffith, and Jacob and Monod. It was Jacob and Monod. Frederick Griffith, I told you in the previous classes, he did experiment on in the search of genetic material with what we call the Griffith's experiment with the mice and the bacteria pneumococcus. Okay. Hershey and Chase did another experiment with the bacteriophage. So Jacob and Monod were the scientists who proposed this model of lac operon. Okay, lac Z gene code for the enzyme beta-glucosidase, right? Not polymerase. It is the beta-glucosidase, not transcript. So it is the beta-glucosidase. Okay. So you know that this is the enzyme that is involved in the breaking down of the lactose molecule into lactose and glucose. So this was all about the lack of regulation of gene expression. So regulation of gene expression, this was about the prokaryotic cell. Taking the example of Escherichia coli. Okay, so it's not the time when this Jacob and Monod did this experiment. It was not easy. Okay, whatever the available resource and the technology that they had at that time, whatever the scientists have done experiment, it is really very is commendable, and it really is very surprising. Okay, because for their effort, and we must applaud their for what work they have done. Due to the work of these scientists, we are able to understand the different phenomena and the procedure, the process. Okay, and due to this, we are also able to understand the underlying cause of the many diseases, and also we are able to find the different solution and the treatment by studying the different functioning or with the role of different the structures, molecules, genes that is present in our body. Okay, so this was all about the regulation of gene expression. Okay, so thank you for joining and listening today. Tomorrow again we'll meet at the same time. Okay, so till then, goodbye, take care, and we'll meet at tomorrow at the same time. Okay, so bye for now.